So my name is Dory Abelman, and I work with Trevor Pugh, Suzanne Trudell, and Keith Stewart at the Princess Margaret Cancer Centre in Toronto. And we're trying to make less invasive ways of detecting minimal residual disease for people who have multiple myeloma. And one of the ways that we're doing that is by looking at cell-free DNA. What we do at the beginning is we take a bone marrow aspirate right at diagnosis. This is done alongside other tests that a patient would receive at diagnosis. And we take DNA from that and then sequence it by whole genome sequencing. And from that, we can get thousands of mutations. We typically get about 10,000 mutations that are somatic, so unique to the tumor, and not found in the patient's other cells. And then we follow up by looking for those mutations over time in the cell-free DNA through treatment. And we're also doing whole genome sequencing on the cell-free DNA. And the goal of this work is to give patients who don't want a bone marrow aspirate another option for detecting their minimal residual disease, which is really important for their risk stratification. And some other benefits is not requiring patients to come in to a major cancer site to get their sample drawn, because a normal blood draw we could do anywhere. We could do it at Life Labs, any clinic, whereas a bone marrow aspirate requires a bigger center to be done. Another benefit of looking at the blood is we're able to capture DNA from all the sites in the body. So let's say that there's a tumor. So myeloma is typically very patchy. Let's say there's a collection of cells that's away from the site being profiled from the bone marrow aspirate. Those aberrations might be missed when we're doing serial checkups. But in the blood, we're able to get more of that extramedullary and patchy disease. So what we did was a preliminary analysis where we took patients on a study called the M4 study. It's a study of about a dozen sites across Canada, newly diagnosed multiple myeloma patients, all receiving the same standard of care. And we ran this test on a subset of them, in addition to a whole bunch of other standard MRD detection tools. For example, multi-parameter flow cytometry, adaptive clonoSEQ, which is sequencing of the immunoglobulin region. We also had PET scans done, and EZM, which is another new MRD detection technology that uses mass spectrometry on the M protein in the peripheral blood. And we compared the results of all of these tests together. What we found was that EZM, the mass spectrometry technology, had the most MRD positives. Actually, all of our patients were MRD positive by EZM right after their stem cell transplant, at that 100 days post-transplant time point. We had the second highest positivity rate for our cell-free DNA mutational signatures method, and then lower rates for flow cytometry and clonoSEQ. When we followed up with these patients again, after 1 year of lenalidomide maintenance therapy, we again had the most positives by EZM and a very high concordance rate, over 80%, with our mutational signature cell-free DNA method. Both the mass spectrometry of the peripheral blood and our cell-free DNA whole genome sequencing found a lot more MRD positive cases than the traditional methods like multi-parameter flow cytometry and adaptive clonoSEQ. And we think that's because we're capturing more extramedullary disease, or disease outside of the site of just a regular biopsy. So we're very hopeful from these preliminary results, but we're looking to make this even better, trying to improve the sensitivity of our tests, and also validating this finding on a broader and larger patient cohort. Another advantage of using mutational signatures approach for detecting minimal residual disease in cell-free DNA is that it can work for all kinds of patients. So EZM is currently limited to patients who only have a normal heavy chain myeloma M spike. So it won't work on light chain only patients or non-secretory patients, whereas the mutational signatures will work on both. So we're hoping that this method will be especially helpful for patients who wouldn't be able to benefit from EZM, or who live far away from a major center and don't want to have a bone marrow aspirate done for the other conventional methods.